Hi everybody, we're going to take a look at human sex-linked alleles today. Hi everybody, we're going to talk about human sex-linked alleles today. Pause the screen for learning goals and success criteria before you move on. American biologist Thomas Hunt Morgan did research relating to genes that didn't assort independently on the fruit fly. So with these fruit flies, he chose them because they were relatively inexpensive, reproduces quickly, and shows traits that can be easily characterized. And in his research, he saw that there were some things that are different than Mendel's results with genetic inheritance. He took a white-eyed male and crossed it with a normal red-eyed female. All of the F1 generations that came out all had red eyes. This showed that normal red eyes are dominant to white eye mutation. So very similar to what we did with one trait crosses. One of them was dominant and the other was recessive. Now, if you do a purebred cross of both, you're going to get all heterozygous, but they're all going to show the dominant trait, where the allele for the recessive is going to be hidden. So the difference here is that when Morgan took a male and crossed with a female from the F1 generation, the results were slightly different than what Mendel had shown. So all of the females in F2 generation had red eyes, and all of the males, actually half of the male had red eyes and half of them had white eyes. So in this illustration here, this is our parents that Morgan took. So we have red eye female and white eye male. He crossed them. We have 100% of the results, regardless of gender, they are all going to be red eye. Okay, that's because they're all heterozygous. Now, taking a male and a female here, since they're all the same anyways, take a female and a male, he crossed them, okay, and the results now are different because he has, the females are here, they have red eyes only, whereas you have males, which have a combination. So 50% of the males had red eyes and 50% of the male had white eyes. So the fact that the gene for eye color was connected to gender showed that the gene for eye color is located in the X chromosome. Like human, female fruit flies have the double X for females, and male fruit flies have X and Y. Fruit fly in F1 data show that the white eye phenotype is recessive as it was masked in this generation, so very similar to our 1 and 2 trait crosses. So how did that reappear? The answer goes down to the X and Y chromosomes. We call this sex-linked traits. These are traits that are controlled by genes that are in either the X or Y chromosome. These are the genes that determine sex, and they're linked to those. They are identified by different rates of appearance between males and females. The X and Y chromosomes have very limited amount of genetic information. So the X chromosome has about 2,000 genes, while the Y chromosome has about 100. While the most important job that they have for the X and Y chromosome is for sex determination, there are also genes that are attached to it, and that's how we get our sex linked. Now, females will have 2 copies of the traits that are expressed in the X chromosome as opposed to 1 in females. And traits related to X chromosomes are called X-linked, where traits related to Y chromosomes are then called Y-linked genes. When we look at sex-linked traits, the allele for sex chromosomes is shown as a superscript to an X or a Y. Now, that's because we're only looking at sex-linked. If it's sex-linked, we have to be able to tell what sex it is, and from our crosses, we have to be able to indicate which sex is being crossed and what alleles are with those. So in this situation with the red eyes, we know that it is X-linked. It is an X-linked gene. So that means the allele will only go with the X chromosome. So let's take a look at this chart here. In this chart, all of the genotypes here, we have 5 possibilities, 3 possibilities for females and 2 possibilities for males. If you look at X and Y that are indicated here, because it's an X-linked gene, the allele will only follow the X and it will not follow Y because it's not linked to Y at all. We know that red eye is going to be dominant allele. For a female with red eyes, they could either be homozygous dominant, so X uppercase R, X uppercase R, or a female that has red eyes with heterozygous genotype, which is X with uppercase R, X with a lowercase r. Now here it's very similar to our 1 and 2 trait crosses where the recessive is masked here and only the dominant allele is expressed. Now if both recessive alleles are present, then there's no dominant allele, then the only recessive allele is expressed here. So X lowercase r, X lowercase r. And then now with males, the difference here is there's Y chromosome, which does not have anything to go with it. So that means our X and our Y are separate. Only X's have the allele that go with it. So males with red eyes will only need to have one allele for the dominant allele, and males with white eyes will only need to have one allele for the trait. In sex-linked traits in humans, many are genetic disorders, and if a disorder is X-linked dominant, affected males pass the allele only to their daughters, and they have a 100% chance of getting that disorder. Now, because if it's X-linked dominant and the male, so let's say the male's XY for sure, right? So let's say it's X-linked dominant, I don't know, for a trait that uppercase R. Now, the daughter is going to get that specific X from this male, and that X will have the dominant trait. So that means as it goes to the next generation, if this is dominant, that means the daughter who will take that X from this father will have the dominant allele in their genes. So regardless, the daughter will show 100% of that specific allele. Now, females can pass an X-linked dominant allele to both sons and daughter, and they both will have this disorder. Most sex-linked inherited traits in human are X-linked recessive traits. If we think back to a couple slides ago where we said the X chromosome carries more information, this is also why. While males only need to inherit one allele to be affected, the female must inherit both alleles to be affected. X-linked recessive traits affect more males than females in the family. Here are a list of more X-linked and Y-linked recessive and dominant conditions. Some of these are recessive, and we just said that most of them are X-linked recessive. So this whole list here, X-linked recessive, so it's all based on the gene that is attached to the X chromosome. Here's an X-linked dominant, and here's a Y-linked. So Y-linked means as it is an allele that travels through generations, it only is attached to the Y chromosome. And that means it cannot be passed on to females. It can be only passed from male to male. Let's take a look at CVD, color vision deficiency. So these are individuals that are affected. They have varying degrees of difficulty in distinguishing between different colors or shades of color. So one form is called red-green CVD. It is an X-linked recessive disorder. They have difficulty distinguishing between shades of red and green. Take a look at the picture here. The one on the left is what a normal person without the CVD disorder is going to see, and then the one on the right is going to be what somebody with red-green CVD is going to see. Note that it's very difficult to see to begin with, but it's the shades of red and green that is very difficult to distinguish. Hemophilia is another example. So that's a condition that affects the body's ability to produce proteins involving in blood clotting. People with hemophilia can suffer serious blood loss from simple cuts and bruises. It is a X-linked recessive trait that affects more than 3,000 individuals in Canada. It's often referred to as the royal disease because back in the days in Europe, Queen Victoria was a carrier who passed that allele on to some of her offspring. Okay, let's get down to our Punnett squares here with a sample problem. It says to determine the probability that a woman who is a carrier for hemophilia and a man without hemophilia will have a child with hemophilia. This should be have, not had. You can make that change there. Now, hint that you got to determine genotypes and phenotypes of offspring to determine if they have hemophilia. So we're talking about hemophilia. I know this is not a dominant trait, but what we're going to do is, because it is a sex-linked allele, and to simplify this, we want to make sure that we can see this is hemophilia. So for the purpose of this question, we're going to use H as the symbol that represents hemophilia. And because it's recessive, that means if it has a lowercase h, that is the actual trait. Uppercase H is going to be a normal blood. So let's get started. We have male and female, let's put those chromosomes in first. And we have a woman who is a carrier, means she's carrying one allele that has hemophilia and the other one doesn't. So as we were saying, that you use lowercase h as the allele for hemophilia, that means uppercase H is going to be normal. And for the male who does not have hemophilia, is going to be an uppercase H here. Now, once we have their genotypes, we can actually cross them. When we cross them, it looks like this. All right, we have 4 possible answers here. So the first daughter is going to be homozygous dominant. So this means no hemophilia. This one right here, so the second daughter is heterozygous. So what shows is going to be normal blood. This male here has the dominant allele, so nope. So the only one that we have here with hemophilia is this one that shows, this male that shows a recessive allele for hemophilia. So there's a 25% chance that their child is going to have hemophilia, where the rest of them will not. They will have normal blood clotting blood. Okay, so what you're going to do is, there's some homework questions. Please take a look at them. The answers are going to be posted on to the genetic problem sets as well. So make sure you take some time to answer them and test it out. Okay, if you have any questions, make sure you ask. Send me through an email or with the Q&A, or if you do end up catching me during office hours, please do so as well.